My name is Karin Wayne. I'm a genetic counselor at Geisinger and a member of the ClinGen Education Workgroup. This presentation will provide a brief introduction into the spectrum of genetic variants and how to identify variant spectrum information when considering a variant interpretation. Understanding the variant spectrum of a gene is an important step in variant interpretation. It allows us to become acquainted with the types of genetic variants that are known to cause a disease within a specific gene and what the mechanisms of disease are. In other words, why a variant causes a disease. We can also learn if there are variant types that are often seen in the general population in presumably unaffected individuals, which can indicate that a variant is less likely to cause disease. The location of a variant within a gene can be important to consider. And finally, the types and locations of variants in a gene can be associated with different phenotypes or even distinct disorders due to different mechanisms of disease. So thinking about variant spectrum is about knowing your gene. It is important to be clear about the terms used to describe different types of genetic variants and the expected consequences of each. Some types, such as nonsense, frame shift, splice site, and large deletions, are often referred to as loss of function variants, since they are predicted to lead to complete loss of gene product through nonsense-mediated decay. Other variants, such as in-frame deletions or duplications, and missense variants, are generally expected to result in an altered protein that may still be stable. Variants which are not expected to change a protein include silent and intronic variants, though these can impact splicing. Finally, whole gene duplications are associated with disease if there is evidence of triplo sensitivity for that gene, which is not the same as gain of function. For a review of variant nomenclature, please view the specific webinar available on the ClinGen YouTube channel titled Introduction to Variants and Nomenclature. The spectrum of variants that are pathogenic or likely pathogenic for a disease is best learned from data from clinical populations. The published literature is a primary source for this data, but literature searches can be challenging. In addition to PubMed or other literature searching, curated literature resources can be helpful to identify relevant literature. The DBD, or Developmental Brain Disorders Gene Database, OMIM, HGMD, or the Human Genetic Mutation Database, and Gene Reviews are all examples. While OMIM, HGMD, and Gene Reviews are general resources for many types of genetic disorders, the DBD database is specific to genes associated with neurodevelopmental or neuropsychiatric disorders. An important caveat to mention is that these resources may not contain every reported variant or variant classification, and variant classifications might reflect the author's perspective at the time of publication rather than current variant interpretation guidelines. For additional information about literature searches for sequence variants, view the webinar on the ClinGen YouTube channel that specifically focuses on this topic. Gene-specific databases are another very useful resource to understand variant spectrum and disease mechanism. The databases shown here are just a few examples. These databases can be made available from clinical laboratories through testing affected individuals or from research efforts. While sometimes details are lacking, they can also be helpful by providing a primary literature reference. Here is an example of the ENG gene database from ARUP laboratories. This graphic gives an overview of the types of variants in the database and the proportion of variants with each clinical classification. The database provides a complete list of all variants, including the location, variant type, nomenclature, classification, how many times it has been observed, and how it was identified, including publications. You can scroll through the data or sort by the headers. Sorting can allow you to see the types of variants generally considered to be pathogenic or likely pathogenic. Scrolling through can also help identify other reported variants that might impact the same amino acid as your variant of interest, which is an important criteria in variant interpretation. Large-scale data sharing has become an extremely valuable means of pooling data across clinical laboratories, institutions, and databases. ClinVar and Decipher both represent publicly available databases of clinically obtained genomic variants, both sequence level and structural variants. Remember that the classification of a variant in ClinVar, known as an assertion, comes from the individual submitter, not from ClinVar specifically. Now that we have considered the use of clinical or patient-derived data in evaluating variant spectrum, we can touch on how large general population data can be helpful. Just as some variants of a particular type or location in a gene may be more concerning, population data can indicate whether a variant type may be less concerning for disease. Large population data sets, like gnomAD shown here, provide a statistical analysis comparing the number of expected variants of a specific type to the number observed. This provides a constraint metric that can indicate if a variant type appears to be tolerated or not. In this example, the MBD5 gene is constrained for loss of function variants, but not for missense variants. For additional review of population data and how it can be useful in variant interpretation, please view the other webinars on the ClinGen YouTube channel that specifically focus on this topic. The location of a variant within a gene can be an important consideration. Some genes have hotspots, specific regions where disease-causing variants are found. This is often associated with a specific disease mechanism, such as gain of function, and specific variant types. Some types of variants are usually predicted to cause loss of function through nonsense-mediated decay, such as nonsense variants. However, if these variants occur near the 3' end of the gene, degradation of the gene product may not occur. In these situations, there is a stable protein product, which is abnormal in length, but may or may not be able to function in a normal way. As we saw in the previous slide, population data constraint metrics can inform variant interpretation. Currently, exon or region-specific constraint metrics from population data are being developed, and in the future will likely help us determine regions of genes that are particularly intolerant to variation. There are also examples of genes where different disorders or phenotypes are associated with distinct variant types. This can be due to different disease mechanisms. For example, in the SCN2A gene, missense variants believed to cause a gain of function are associated with seizure disorders, while loss of function variants are usually associated with intellectual disability and/or autism spectrum disorder, often without seizures. In other circumstances, a variant type may be associated with milder clinical presentations due to partial protein impairment rather than complete loss of function. This concept is referred to as genotype-phenotype association. Finally, I would like to bring your attention to ongoing gene and variant curation efforts within ClinGen. At www.clinicalgenome.org, you can view the many expert panels and the outcomes of these efforts. There are currently over 1,500 gene-based curations and over 10,000 variants curated and submitted to ClinVar by variant curation expert panels. You can either scroll to find your gene of interest or use the search field near the top to see what curation may have been completed. And the ClinGen dosage sensitivity map can be particularly helpful in understanding disease mechanisms, such as haploinsufficiency and triplo sensitivity, and to learn if any specific genes within a large copy number variant might be of particular clinical relevance. This resource is another useful starting point to direct you to key literature regarding a gene or genomic region. Specific webinars which further describe these curation efforts are also available on the ClinGen YouTube channel. I hope this has been a useful introduction into the importance of genetic variant spectrum for variant interpretation. Please visit www.clinicalgenome.org for additional educational resources and to learn more about ClinGen initiatives. Our funding sources are listed at the bottom. Please feel free to contact ClinGen by email at clingen@clinicalgenome.org with questions or feedback. Thank you.